Great. Well, welcome everybody to the February Cells to Society seminar. Our theme today is food safety, and we have two wonderful talks lined up, one from Elizabeth Newbold on NECAFS, the Northeast Center to Advance Food Safety, and one from Dr. Andrea Etter on Listeria monocytogenes in artisan cheese. To get started, I do want to do a little bit of background on the Cell Society seminar. Our purpose here is to provide a cross-pollinating forum for sharing research, education, and extension across CALS and even across UVM university-wide. Got a great team that tries to pull these together and organize them, and we're getting further and further out with our planning each time we get together, which is a nice way to reduce stress on all of our parts. Appreciate Andrew May and Hans Estrin being a part of the team pulling these together, and Kathy Yandow, Tina Haskins, and Joanna Cummings for helping us promote them across the college and the university. You can always find information about the Cell Society seminar at go.uvm.edu/cells2 society seminar. We do have a YouTube channel as well, where we post recordings of the seminars for viewing at any point in time at your convenience. You can find that at the same place. So these are, this is one seminar in a 2-month seminar sort of cycle that we're in. And then at the end of, in the third month, we hold what we call quarterly out-of-the-box brainstorm sessions. And these are not seminars. These are intentionally crafted to be held in breakout rooms where the themes that have been discussed in previous seminars are used as an opportunity to talk about potential collaborations or projects, or just to share current work in a little bit more informal way and among a larger group of peers focused on those topics. So we have one more Cells to Society seminar following this one before our next brainstorming session. Today's theme, as we mentioned, is food safety. In March, we were delighted to be focusing on microbiology and molecular genetics, or MMG, as some of us call it. And in the month after that, in April, we'll have our out-of-the-box brainstorming session on April 13th. So as I mentioned, you can find more information about the seminar and our previous recordings at the main seminar page, Cells, the number 2 Society seminar. So with that, I'm going to turn it over to Elizabeth Newbold for our first presentation today. We'll do two presentations, and then we'll have combined Q&A at the end. Elizabeth, take it over when you're ready.
Okay, thank you. I will share my screen. Are you seeing it in presenter mode?
It is. Yep.
Okay.
Perfect. Thank you.
Okay. All right. Well, I would like to thank Chris for the invitation today. My name is Elizabeth Newbold. I'm from the Northeast Center to Advance Food Safety, and with Chris Callahan, we co-lead the Northeast Center. And I'm just going to first give a quick overview of what NECAFS is and how it came to be, and then I'll talk about the work that we do. So NECAFS was funded in 2016 in order to support the rollout of the Food Safety Modernization Act, or FSMA. And FSMA is a rule that was passed in order to help prevent foodborne illness outbreaks throughout the food supply. And within FSMA, there are 7 rules, and NECAFS specifically focuses on the 2 rules that focus on produce safety, which is raw agricultural commodities, and preventive controls. And that's produce that goes through some step of processing. And so, as I mentioned, in response to that rule in 2016, the USDA and FDA funded 4 regional centers, ours being the Northeast Regional Center. There's one in the North Central region, that's through the University of Iowa. There's a Western Regional Center through the University of Oregon, and a Southern Regional Center through the University of Florida. And each of the regional centers takes sort of their own approach to rolling out food safety support. And I'm just going to quickly walk through the objectives of the Northeast Regional Center. And so what we do is we enhance produce safety and preventive controls, training, education, and outreach among small and medium-sized farms and small processors in the Northeast. And so I kind of want to point out that the language here of small and medium-sized farms and small processors in the Northeast. So our focus is really on the small producers and processors who are impacted by the produce safety rule. They might not be covered by the produce safety rule, but they are feeling pressure either from the market or from consumers to adopt practices on their farm in order to produce a safer food. And so how we've done that in the Northeast is by forming what we call our network. And what that is we bring together the educators and the regulators across the 12 states within the Northeast region who support implementation, education, and outreach to producers and processors. And we discuss the common issues that those educators and regulators are seeing on the ground. And we think about how we can collectively together help to address those challenges that our region's producers and processors are facing. And the way that we do that is by forming a collaborative network. And you can see across the bottom our objectives. And this is really quite intentional. You can see that they're arrows connecting them. And the goal of that is because we are taking an intentional approach toward creating connections among our region's educators and regulators in order to develop trusting relationships over time and ultimately result in sustaining good work. And so the goal is to enhance produce safety, preventive controls across our region, hitting small and medium-sized farms and small producers through joint action. And I want to share with you this collaborative spectrum. This is particularly meaningful to me in the work that we do and the work that NECAFS has done since 2016. And so you can see on here, there's this spectrum where on the left-hand side, you have sort of independent work and activity where people are working within silos or working isolated on issues. And as the spectrum moves to the right, the language becomes more collaborative. So you move from coexisting to communicating where you start to share information, you start to share resources, to cooperating, which is a much more intentional, discrete activities, working on discrete activities and projects. And then you start to move into these more integrated forms of collaboration where you're coordinating and actually collaborating, where your activities are depending on each other. And I want to just add here that it's a very, this is what's sort of important to me, is that when you're on the left and you're not working together with others on this spectrum, you really have independent parallel practice, which is what we saw sort of in the early days of food safety across the region, where people were working independently in their states or in their communities, but they weren't sharing their knowledge or their areas of expertise with others in the region. And so NECAFS has really been very intentional about bringing all those various forms of expertise together so that now we're further along in that spectrum and we're actually collaborating, which means that our work is interdependent. We are interdependently relying on each other's point of views, perspectives, and expertise in order to collectively work on co-action. And so what that looks like is NECAFS actually starts to bring people together, and at a very large annual meeting, we talk about the areas of need for the region, and then we form project teams from across the region in order to work on those identified areas of need. And so I'll now talk about some of the work that we've actually completed over the last several years and what we're working on. So first, I want to point out the team. You'll see here Chris and myself, but we also have 3 full-time staff, Annie Fitzgerald, Sean Fogarty, and Anna Lowald, and they are all research specialists and focus on both preventive controls and produce safety and helping to actually implement the activities that our network has described. And as I jump into sort of the actual work that we've been doing, one of the areas is produce safety and hydroponic and aquaponic operations. And so if I take a step back before I jump into the activities, one of the things that we noticed early on was that all of the people who work within our region, the educators and the regulators, they are pretty much fully committed to work that they're doing at their university or at their organization. And so to add on sort of additional work that was regional in nature, it just wasn't happening naturally. And so NECAFS committed about 3 years ago to hiring staff specifically to execute the deliverables that group. They were very happy and had time to come together to talk about the work that needed to be done. However, they didn't have the time to actually do the work themselves. And so what we've ended up doing is we've formed what we call work groups, which function a bit like advisory groups, but they're much more heavily involved in the work. And they oversee the work. They contribute through meetings, feedback, collaborative brainstorming sessions in order to help inform the actual work that's executed. And then the staff at NECAFS sort of leads the actual day-to-day implementation of the work, and we adjust, make edits to the work based on the feedback we receive from those work group members. And we've found that to be a very effective way of actually executing the projects that everyone in the region has agreed are needed. And so we've adopted that model across much of the work that we do. And I'll start here with this first one. So there was a clear need that for this very specific niche audience of aquaponic and hydroponic growers, there weren't many resources in food safety available, and that we needed to start to create resources that introduce the topic of food safety to this audience specifically, because they weren't able to see themselves in sort of general food safety material and needed something that referenced those types of growing systems more directly. And so about 2 years ago, we began the process of creating these series of fact sheets, and we'll be releasing them later this year. And they dig into specific sections of the rule, but through the lens of aquaponic and hydroponic operations. As a result of that work, we received so much positive feedback and interest in those fact sheets over the last 2 years that it became abundantly clear that there was a need for a more formal and extensive project that leveraged that early work, but continued the development of resources. And so last year, we saw additional funding from the USDA in order to continue that work. And again, following that work group model, but now moving to a national level. We have a project that's actually led by Chris to develop additional food safety resources specific to hydroponics and aquaponics, but now informed by a national work group. And so that project is just now getting underway. Another thing that we have going on is called the Northeast Produce Safety Research Consortium, and this is something that's just a year into implementation. And this is funding that we received from the FDA, and the FDA was interested in collecting samples, on-farm samples across different cropping systems in order to understand the presence and prevalence of pathogens, viruses, and parasites across different cropping systems. And so over the last year, we've been working with two partners in the University of Delaware and the University of Maine to collect on-farm samples in order to have them tested for various pathogens and viruses and parasites. And you can see here, these are the organisms of interest that were tested for in the first year. And all of the testing is taking place at the University of Maine and at the University of Delaware. And we are working together with those partners to expand this project over the next 4 years. And this is just a very quick summary of what we collected last year during the growing season. And we're using the information from that collection in order to inform our sampling process going forward. And the goal is to both expand the number of collections that we take in the existing cropping systems, but also expand to additional sites across the Northeast as we have more capacity and sort of get a better handle on the work that's involved in this project. Another area that was identified was the need to summarize existing research and extend it to educators and regulators who need that information. And so the produce safety rule is written flexibly and vaguely, and it is specifically done to practice decisions are made based on what science has found around specific areas. And so a lot of the questions we were receiving from the group was, I would love to know what the research says when it comes to pathogen survivability on a tomato that falls on plastic mulch. And it became clear, that's just one example, but it became clear that research is being completed and published. However, there's a sort of a gap in communication of extending those findings to practice so that educators and regulators know how to apply the principles as being found in research. And so we received some additional funding from USDA 2 years ago to specifically look at existing produce safety research, do some literature reviews, and summarize that and publish those literature reviews, and then create educational materials that distill that down and extend it to a more digestible form to those educators and regulators who can then help growers make informed decisions based on what the science says. Because at the end of the day, the growers, the educators, and the regulators don't have time to be going and doing those extensive literature reviews. And so we identified 5 topics sort of being priority areas for the region. We have already completed a lit review on dropped and drooping produce. We are currently in the process of doing a lit review on animal contamination. And then we will be looking at surfaces, pathogen survival, wash water, and water systems in the future. And this is a 3-year project. Okay, so I have 5 minutes left. So I'm right on time. All right, so the other thing I wanted to mention was our preventive controls work group. All the activities I've talked about to this point are produce safety. But one of the very active work groups we have is the preventive controls work group. And what they did very on is they took a different approach. And they said in our region that there's some actually fundamental challenges for rolling out preventive controls to small and very small processors. And it doesn't have to do with the topics within the preventive controls rule, but rather the awareness that the preventive controls rule exists. And so what they've found is that this audience is particularly challenging to reach because very small and small processors don't know that there's a rule that may impact them, and therefore they are not seeking out educational information in order to adopt practices and potentially come into compliance with the rule. And so they have focused a lot, this work group has focused a lot on actually creating and extending awareness of the rule. And as a part of that, they have developed an online toolkit, Processor Food Safety Online Toolkit, which is really a curated tool for small and very small processors who are just becoming aware of the rule in order to guide them to resources that they might need for compliance with GMPs, good manufacturing practices, and the PCHF rule. And so they are now in the process of working with organizations like the AFDO, the Association of Food and Drug Officials, the state's regulatory partners, and others to actually do a much more grassroots outreach to processors that the rule exists. And now that there's this tool that's targeted specifically at small and very small processors to kind of on-ramp them to the PCHF rule. And then one last thing that's actually launching next week is sort of this a produce safety handbook for buyers. And so this is something I worked with Hans Estrin on. This was identified several years ago where there's kind of a communication gap between the buyers, the end market, the grocery stores who are buying produce from produce growers who don't necessarily know the language of the produce safety rule. And to tie into all of this, there are state audit programs that predate the rule that growers sort of conflate the language and start to ask buyers conflate the language and start to ask growers for requirements that might not be relevant to them. And so we went through and created this online toolkit specifically for buyers. And this focuses on the Northeast region, where a buyer can go to this website and interact to learn what food safety requirements a grower may be subject to, depending on the state where they are located. Because, and this is a whole nother layer of complication, the rule, even though it's a federal rule, is implemented and inspected at the state level. So the state departments of agriculture are the ones who are actually enforcing the rule, which means that it doesn't look exactly the same in every state. And so even though it's a federal rule, there may be different requirements depending on how that state has adopted the rule, or if they're working under FDA credentialing or other things. And so therefore it kind of complicates this rollout and that communication between buyers and growers even more. So this last slide just shows that we have other activities going on. And these are some of our sort of early activities where we provided funding for folks who might need to go to trainings. We provide a water lab map that just helps people get connected to where they can do on-farm water testing. And then also we do a lot of pre-post test assessment of knowledge gain from the produce safety grower trainings. And so I think as I sum up, because I have a minute left, my main takeaway in all of this is that what we do is we really respond to the need we hear from our region's educators and regulators, and collectively we discuss and brainstorm how we want to address those challenges in a way that will result in greater impact. And so instead of, if we think back to that spectrum, instead of working independently in silos, we come together to create a network that works interdependently so that we all benefit from our collective work and our collective impact. And so with that, I appreciate your time and we'll take questions at the end. Thanks.
Thanks, Elizabeth. It's no matter how many times I've heard about NECAFS and talked about NECAFS, there's always something new. So appreciate it. Andrea, welcome. Hopefully you're able to, thank you, yep, share. I'm a huge fan of artisanal cheese and have huge respect for Listeria, in particular LM, and I look forward to learning more from you on this topic. Thanks for making this happen, especially on short notice and especially from the airport.
Thank you. All right, so today we're going to talk about Listeria and artisanal cheeses, specifically thinking about raw milk cheeses made on farm. So Listeria is not one of the more common pathogens people are aware of. It tends to be the red-headed stepchild of food pathogens, but it's really important in part because it is literally everywhere. It's pretty much a joke at this point. At the start of every paper on Listeria monocytogenes is something along the lines of Listeria is a ubiquitous foodborne pathogen, blah, blah, blah. Seriously. So when we say ubiquitous, we actually do mean ubiquitous. It's in the soil, it's in groundwater, it's in leaves, it's in the guts of mammals. A certain percent of the population is actually an active carrier without having symptoms. Unfortunately for food safety, it is pretty cold tolerant. It will actually grow at a properly calibrated refrigerator's temperature, so 41 degrees Celsius or Fahrenheit, 4 degrees Celsius. And it is just fine with a certain amount of acid. It is very happy with up to like 10% salt. And so it's really hard to develop interventions for food that are going to keep Listeria from being a problem. The one thing it is sensitive to is heat. So if you're going to cook the food before you eat it, it's going to be pretty safe from Listeria. But if you're not cooking the food, if it's what's considered a ready-to-eat food, it has a potential to cause major issues with Listeria. Its other claim to fame is that it has the highest mortality rate among the common foodborne pathogens. So generally, things like E. coli, salmonella, we're looking at about 0.5% of patients for that illness will die. Listeria, we're about 16% if you're in the susceptible group. What is the susceptible group? We call them the yobbies, so young, primarily neonates, old, and old is very generously sized, so anyone over 65. Pregnant women are susceptible, or more specifically, their fetuses are susceptible. Listeria can cross the placental barrier. And anyone who is immunocompromised in any way has a higher risk of contracting listeriosis. And so when we say immunocompromised, it's pretty much if you'd be at increased risk for complications from COVID, you are at increased risk of listeriosis. It's not a particularly prevalent pathogen. It's estimated to cause about 1,600 cases a year in the US, but it does cause nearly 300 deaths, which makes it the second highest cause of foodborne illness-associated death. And further, it has a 90% hospitalization rate. So if you get Listeria, you're going to spend time in the hospital, almost certainly. So Listeria is a very capable and tricky pathogen. Pathogens that you eat in your food are supposed to stay in your intestines. Listeria is very good at invading the intestinal cells, getting in through the base layer below that, and getting into the bloodstream. From the bloodstream, it can actually cross the blood-brain barrier, which is supposed to keep pathogens out, and get into the brain. It can also cross the placenta, which is why it is a huge issue for pregnant women. Because in a fetus, we see septicemia in the womb. Often this will lead to stillbirth or late-term miscarriage. And if the fetus is born alive, then we'll often see it born with septicemia and leading to either neonatal death or long-term complications, including mental disabilities. For adults, we're going to see septicemia, so blood infections, meningitis, encephalitis. And infections of the brain and spinal cord are extremely deadly. Okay, when we think of these foods that we're normally not going to cook again before we eat them, things like deli meats have long been thought of as the key, and they are still an important source of infection, especially if you're buying them from the deli counter. Sausages are an issue as well. Produce can be an issue. The most deadly Listeria outbreak was actually from cantaloupe. And then dairy, specifically raw milk cheeses, and soft cheeses. Okay, so the artisan cheese environment is unfortunately a pretty happy environment for Listeria. It's there's lots of nutrients because milk is a very good growth medium. And it is, if it establishes itself in the environment, it is pretty resistant to being removed. Work I did in my PhD on retail deli environments, so grocery store delis, shows that if you get Listeria really entrenched in a deli or any other food processing environment, you can try cleaning more often, you can try sanitizing more effectively, and that will probably not touch it. About the only thing you can do is rip the deli apart, scrub everything deeply, retrain the personnel, get them involved in the deep clean, and then it might be successful, depending on how deeply the Listeria is entrenched. So farmstead cheese making operations are a big deal for Vermont and the agricultural economy. And they're also a really good environment to study Listeria persistence because we have a more closed environment, you know, people tromping in and out of it like you would for a deli. It's, you have a lot of nutrients coming in. And for things like artisan cheeses that are maybe aged on one board, there's specific opportunities that are hard to get rid of for Listeria. So when we think about Listeria establishing a persistent presence in an environment, there's a couple of different ways it can do that. Really, the formation of a biofilm is the most important mechanism. A biofilm is essentially bacteria gluing themselves to a surface using sugars and lipids, and in some cases for some bacteria covering themselves with those, and this gives them a lot of resistance to environmental stressors, including drying out. Many sanitizers are not effective on biofilms, and even scrubbing is not guaranteed to remove all Listeria. So this work was funded by the George Walker Milk Fund, and the rationale behind this is really looking at outbreaks that have happened in other artisan cheese spaces. So Volso Creameries was in northeastern New York. And in 2017, they had an outbreak associated with soft cheeses, specifically raw milk cheeses. And the wrinkly rind means they're raw milk cheeses, or sorry, washed rind cheeses, which are even more risky because you're handling them a lot and you're usually inoculating them with a particular solution on the surface that if you use for multiple cheeses and one of them is positive, you now spread it to the others. Listeria outbreaks almost always fall into the small but mighty category. So when we're thinking about Listeria, it's not going to be a lot of cases, but you have 8 illnesses, all of them are hospitalized, and 2 people die. We also have 1 newborn that is born with Listeria. Okay, so this was a multi-state outbreak, primarily in New England, and it was definitively traced back to Volso Creameries. The strain of Listeria that was found in the patients was found in the cheese in the patient's home, and the New York State also found it in 3 wheels of cheese that had not yet been sold. Unfortunately for Volso Creameries, they were actually very much at fault because it was determined that by the FDA that the owners really didn't understand the risks associated with Listeria and making artisan cheeses. So they didn't really have an environmental sampling program, so they didn't have an idea that there was an issue with Listeria in their environment. If you only sample 20 times over a 3-year period, you're not going to have very good detection ability. Unfortunately, there was a pretty high presence of Listeria in these swabs, but they never followed up on it. Now, if you find Listeria in your food processing environment, you need to treat it like your house is on fire. They did not. They also repeatedly found Listeria throughout the facility, even after cleaning and sanitizing, which suggests that there was potentially biofilms because those are the hardest to get rid of. And if you do your cleaning, you do your sanitizing, and there's still Listeria, you probably have a biofilm. And have a happy ending for Volso Creameries. They were shut down. They never reopened. They were a small company. And really, this is the ultimate cautionary tale for anyone working in the ready-to-eat food space. If you are small, as most of our businesses in Vermont are, and you get Listeria and there is an outbreak, you will probably not survive. All right, so I inherited a really massive collection of Listeria monocytogenes from Kathy Donnelly, specifically isolates of Listeria from the artisan cheese space from a study from 2006 to 2008 that found isolates that were pretty similar to each other over the 2-year period. And so that was, the similarities were assessed using older typing mechanisms, and I have the ability to go back and look at that and really figure out, are these the same type of Listeria? Is there actually true colonization? And if so, how are these isolates doing that? Because one of the really big unanswered questions about Listeria is how. Listeria is not a great biofilm former, and there have been a lot of studies done trying to figure out if you have Listeria colonizing an environment, is there something about the isolates, or is it just bad sanitation practices? So the first thing we look at with a set of isolates, and we looked at 31 from this 2-year period out of over 100, is if we grow these biofilms up to the point of maturity, which for Listeria is about 10 days, in high nutrient and low nutrient conditions, what happens to these biofilms when we apply sanitizers? We know that there's evidence that sanitizing biofilms isn't super effective, but there's not a ton of hard numbers. So we can use scanning electron microscopy also to visualize these biofilms, and this has not been done very much. And I love a visual. I love being able to see, what does this biofilm look like? If we see differences, what do these differences look like? And then finally, we are working with the Vermont Department of Health to sequence these isolates. I don't have access to all the metadata on which farm they came from, so I can, with a clear conscience, hand them over to the USDA because I don't have any information, and so nobody can be targeted. All right, so out of the 31 isolates, we chose 6 isolates to do this part of the study. We did some initial screening using 96 well plates for sanitizer tolerance and in liquid culture and for biofilm formation. There weren't any differences, so looking at the Listeria growing in our mature biofilm culture on coupons of stainless steel, which basically a coupon is 1/4 inch in diameter bit of stainless steel, same type you'd find in a dairy or a food processing environment. We see that we have pretty similar rates of Listeria growth on these coupons across all 6 isolates. Isolate 7 is a little bit lower than everybody else, but they're growing to about 100,000 Listeria per, 100 million Listeria per coupon if you give them lots of nutrients. If you give them a poor nutrient scenario, which might be like a corner of a dairy, we see that there's there are a lot, it's more like 1,000,000 isolates, 1,500,000 bacteria per coupon. So there are differences. You get more biofilm if you give them more nutrients, unsurprisingly. And then we would take these coupons, we'd rinse off the sanitizer, and rinse off the broth, and then we'd add them to a vial of sanitizer for the appropriate time, put them into a solution that would deactivate the sanitizer, and then remove the bacteria using glass beads. So when we look at this, we're actually looking at a decrease. So how much does applying sanitizers affect the amount of Listeria on a coupon? So the darker bars are the nutrient-rich scenario, and we see that if you have biofilms grown in nutrient-rich conditions, they across the board do better when hit with sanitizer than isolates grown in nutrient-poor conditions. Even though this is a normalized scenario, so it's not just that the nutrient-rich ones have more bacteria to begin with. They also don't die off as quickly if you hit them with sanitizers. But it's important if you're thinking about places like a drain or a food contact surface, which is getting a lot of nutrients from cheeses and milks. We want to see a 5 log reduction, and even for our nutrient poor conditions in the quaternary ammonia compound, we are not seeing more than about a log 2 reduction. So very ineffective for quaternary ammonia compounds, things like Lysol, essentially. If we hit it with bleach, we see that the scenario is a lot better, but we're still not hitting 5 log reduction. We're getting maybe 4 for isolate 1, and if we go all the way down to isolate 29, we get a lot less, and that's for nutrient poor. Again, these biofilms do better. If we look at nutrient rich, we are getting less than a 2 log reduction, even with bleach, and bleach is extremely effective as a sanitizer. So we did all of this imaging, and we wanted to see what does Listeria look like on stainless steel. The first thing you notice about stainless steel is it's not actually smooth. If you stick it under a scanning electron microscope, there are all these little niches. So all these cracks, and Listeria loves these cracks. So in this hole right here, we see a ton of Listeria clustered, but otherwise it's pretty smoothly spread across the surface. And that's what we see overall for our nutrient-rich scenario. Nutrient poor, however, tend to cluster together into clumps. They're not evenly spread across the surface. They form much more of a 3D architecture. And I'm not sure if this is an artifact or if this is actually real, but these isolates look like they're collapsed, or these bacteria look like they're a bit collapsed in the middle. Okay, so this is without being hit with sanitizers or anything. If we hit coupons of stainless steel with Listeria all over them with sanitizers, we see that there are still a lot of bacteria left after hitting it with Lysol-like compounds. There are a ton of Listeria still in this little hole in here, and they look fine. Outside the hole, they also look fine. There's fewer of them than in the previous picture, but there's still enough. Nutrient poor, it's much more scattered. This is much more zoomed out, but there you can still see occasional happy-looking Listeria. Bleach is a little bit more what we want to see. We're definitely seeing a lot fewer bacteria. We see a fair amount clustered in imperfections on the surface, and they look fine. So bleach is a really big oxidizer. It should just destroy the surface of these bacteria and they should look terrible. They don't. Even in the nutrient poor scenario, where they generally are more sensitive to bleach, we still see plenty of Listeria that look happy. And again, these are clustered into a surface imperfection. Isolate 10 by far shows the most robust biofilm formation. This is actually a lot closer to what we might see in Salmonella or E. coli. So there's a ton of isolates all clustered together. There's some three-dimensional development. There's not a lot of slime layer, but they're definitely growing together and probably communicating with each other. Nutrient poor is different. This one is not particularly robust in nutrient poor conditions. If we hit it with quaternary ammonia compounds, we see a lot fewer isolates or bacteria on the surface for nutrient rich. And in nutrient poor, we actually get our first glimpse of extra polymeric substance, or that slime layer. And these bacteria that are surviving are clustered more into these sections with slime. Unfortunately, if we hit it with bleach, we are still seeing plenty of bacteria. This is more than enough bacteria to get you sick. And we see that it's a lot more spread out than if we look at the first picture. The clumps are still, again, in those surface imperfections. But there's plenty of Listeria that look completely exposed to sanitizers that are still looking happy. At least a lot happier than I think they should be looking. In nutrient poor, we can see some suggestions of cell debris, but also some clumps of Listeria that have survived bleach. So, in summary, if we look across all of these isolates, this is 3 of the 6, the patterns were pretty consistent overall. We see that biofilms formed with high nutrient condition or low nutrient conditions are less dense overall, but they tend to be more likely to have visible extrapolymeric substance, or that slime layer. Nutrient-rich biofilms tend to be more consistently spread across the surface and are less likely to be complex and have three-dimensional development. However, it doesn't seem to make a huge difference. Whether or not you have that complexity that you'd see in something like Salmonella or E. coli, just being attached to the surface seems to change the isolates somehow, that they are more resistant to sanitizer. It doesn't matter if there's other bacteria nearby, like right next to them. It doesn't matter if there's visible exopolymeric substance. These bacteria are survivors if they get an opportunity to attach to the surface. So the first 2 isolates I showed you, isolates 9 and 10, are from a floor squeegee and a fill hoop. So something you pack cheese into. And these ones are generally going to have less, maybe less water moving over them. Squeegees sit in buckets, often with sanitizer. So it's perhaps less surprising that they don't respond very well to sanitizers. But they also tend to form biofilms better. Okay. What are the takeaways? Listeria is still a huge problem. We don't have great answers. We know that if you get it in there and it gets established, you are going to have a lot of problems because it's going to be really hard to get rid of it, short of tearing up your floor, tearing out your walls, tearing out your equipment. And you are going to just keep having issues again over time. We don't have great answers. We're still working on this. There's a lot we don't know. Listeria does not usually form biofilms by itself. Matt Wargo and I are working on a multi-species biofilm model, which makes these biofilms look sad. And I cannot wait to see what it looks like when you add other bacteria to the mix and get something that's more robust. So a lot of need for research, and this research is really vital to the continued survival of Vermont's artisan cheese companies. If they get an outbreak, they will probably not survive it. There are a few companies who do, but it's really easy to just die off, whether because you're getting hit with a lot of lawsuits and liability, or because you're like Bolto Creameries and you never had your act together in the first place. All right, again, this was funded by the George Walker Milk Fund, and I'd like to thank my lab group. So we've had a bunch of people work on this. So Emily Fourier was the original grad student on this project. She now works for Green Mountain Keurig. Spent a couple of years working at Children Farms as a cheesemaker. Laura Cushman did an MPH at UVM after finishing up her undergrad and work in my lab. Aislinn Gilmore is now doing a quantitative epidemiology program with Dartmouth. And Eurydice, of course, is in food systems, is continuing the work. And with that, I will take any questions.
Thanks, Andrea. Fascinating work.
Thank you.
I'm gonna start with the first question we have in the chat box. Maura asked, Maura, if you want to unmute and ask your question, feel free.
Yeah, thanks, Chris. My question was for Elizabeth. I was curious if NECAFS, if I said that right, works with those in the charitable food system, like gleaning operations or school gardens, because a lot of that food is touched by many different hands that might not be aware of the information that you all have. And I'm just curious, if you work with them now or have any plans to do that in the future.
Yeah, that's a good question. And so when I said educators, I should have expanded to say that includes university extension, but it also includes community-based organizations, which that's where you tend to get a lot of the type of audience you were just referencing. That being said, we have a long way to go with the community-based organizations because we have a lot who are part of our network and we work with them and they're very integrated into that, but it is an area that comes up often because those individuals they're not visitors, right? Like they're coming, they're touching produce, therefore they become personnel. Even though they're not paid, they're unpaid, but they're interacting with produce or contact surfaces and they're touching them. And it does increase the layer of education that they would need, not just to be aware that the farm has food safety practices, but they themselves should be receiving practice, food safety practice training so that they know what they should be doing in order to protect the food they're touching. So the answer to your question is yes, but we still have a long way to go because those organizations aren't necessarily as connected to extension as others are, and it is an area that we are continuing to do additional outreach to.
Thanks, Elizabeth. Thanks, Maura. One of the other things that NECAFS does is helps to prevent reinventing the wheel. So if there are resources or educational programs developed someplace else in the region or even in the country that are particularly relevant to gleaning organizations, for example, Oftentimes what we do is serve to connect the local need with the existing resource that might be somewhere else and try to help tailor it where needed, but trying to make people more aware of what already exists oftentimes. Hans has a bunch of questions, wants to know how you interpret those electron microscope scans. Hans, you want to pipe up and ask your question?
Sure. Yeah, thanks. That was fascinating. And yeah, just looking at those scans, when you see a particular bacteria, do you know it's alive? I mean, in that biofilm, can a bacteria sort of stay physically in tune but be knocked out functionally? And are there other ways to kill Listeria, like dry heat, for example?
So you could kill it with dry heat. It's a little tricky in the processing environment. So I can say that there are a lot of bacteria of Listeria that are still alive after the biofilm, because these are the number of we were able to grow up. These log decrees were based on growing the Listeria back up on media after hitting them with sanitizers. So we know that there are a lot of bacteria left that can still recover.
Thanks. And then Elizabeth, I had one which maybe you're typing an answer to. But yeah, just like being part of NECAFS, you know, I love how the organization has evolved. And I think from the early days, we've definitely kind of gone into some new territory. And I'm wondering, you know, if you take that looking ahead again, 5-10 years or whatever, do you have a gut sense of that? Yeah, we're going to bust into some other new realm of collaboration, or do you see this as a sort of mature collaborative form that is going to last for a while?
Hans, Chris and I talk for hours about that without having an answer. So I guess let me frame it in that we are fully soft funded and dependent on our funding from USDA. And so the work that we do is obviously tied directly to the objectives and activities outlined in the proposals that fund us. So I guess that's our guiding principle for now, and assuming that funding stays to sort of, let's say yes, like we exist in 5 years. I don't see why we wouldn't, but what does that look like? To your point, I think we are constantly pushing the region and the collaborative work that we do, because what we've seen is it's an evolution, right? Food safety implementation is an evolution, and the rule is here, but before that there were gaps, there were voluntary standards that growers were adopting. And as there's now a rule, there's more marketplace pressure, we are getting more and more questions that like the work that Andrea is doing is fascinating to me, because you've got cleaning and sanitizing is a huge issue within everywhere, and layers of complication to that. Right now, we're just trying to get people to clean and sanitize. What does that mean when you start trying to consider what's your biofilm? And are you talking about biofilm inside of a brush washer for carrots, or are you talking about a biofilm in a cheese plant? And those are obviously totally different things, and they require different approaches. And Andrea, she mentioned the science isn't there. And so I think what Chris and I are constantly doing is saying, it's not good enough to just say, let's do another PSA grower training. Yes, those things are needed, but if we're really going to move the needle on food safety, we have to continually push our collaborative work to consider all of this evolving knowledge and the evolving pressures that our growers and processors are feeling from both regulatory partners and market, the market. And so I guess to answer your question, yes, I see us looking very similar in 5 years, but kind of striving to continue to get into those nuanced questions and understanding of how do we help our growers truly both make practice adoption choices, but also start to take action that's truly getting at what's causing foodborne illness outbreaks within the, within our communities. So, I mean, so that's my answer. I mean, yes, but continue to push.
Thanks so much.
Other questions?
I had a question for Andrea. I find this biofilm thing fascinating. Like one of the things that in the research that we're like looking at literature that we're looking at sort of in this collective literature review is sort of survivability on different surfaces, and that looking, I mean, you had stainless steel.
Yeah.
Have you looked at, are you looking at it on different surfaces? Wood, other surfaces that we commonly see in these operations?
Yeah, that would be good to do. Wood is tricky because you're probably already going to have some, it's hard to sanitize. I'm actually working with some cheesemakers right now on figuring out what to do when they find Listeria on species on wood boards and like what's the best way to move forward. Because they were boiling the boards, and that creates other issues with cracking that could actually make it more hospitable to Listeria. So it's complicated is the short answer. I have done plastic and I've done stainless steel, but there's a lot of different surfaces that could be looked at. In general, the more porous or imperfect the surface, the better for Listeria or any other bacterium.
Which leads me to kind of my next sort of 10-year question for the food safety work I do is, for, single surface with a set of characteristics, single species, right? Okay,
Yeah.
we can kind of model that and understand that. I'm really looking forward to the work you and Matt are doing in terms of this sort of more community, microorganism community level work, and how does that competitive environment impact measures that, such as sanitizing or cleaning and biofilm reduction.
I mean, we know that certain bacteria are going to be antagonists, like lactobacilli tend to work against Listeria. But it's so complicated because even if you do like metagenomic sequencing of a particular environment, like recreating that in the laboratory may not be possible. So.
Not to mention recreating it in practice, right?
Yeah.
Like,
It's not something to be regulated. But you think of all the thousands of years of artisan cheeses that where they never even washed or they rinsed the things out, and it's part of the whole like ecosystem of microbial ecosystem.
Right. Yeah, it's a very complicated and tricky environment. So.
I mean that, and just real, I guess one. And does anybody else have another question? Because if not, I have sort of almost a pedagogical or cultural question, which is so yeah that thought, Hans, like we run into that all the time. We grow carrots in the ground, and you want us to keep the ground, the soil, away from everything else. Like this doesn't make sense to me. Or like we use wood to age cheese, and you want us to use stainless steel. How are you running into that, Andrea, in your work?
Kathy Donnelly did a ton of work on the on cheese boards and cheese board safety. And I know her position tended to be that it was pretty safe. I'm not sure I agree with that. I mean, I'm not Kathy Donnelly. I don't have like billions of years of experience with Listeria. But I understand why the FDA is concerned. There's a lot of risk factors. So one of the things I looked at in my PhD was actually the prevalence of Listeria that weren't fully capable of infecting humans and causing severe illness. And unfortunately, that didn't catch on with regulators because there were a couple of cases where these largely avirulent Listeria caused severe illness. And so even though it's like really tiny contribution, nobody wants to take the approach of, okay, does it have a full-length internal and aging? And if not, we probably don't need to worry about it, which might make sense, at least in certain environments. It'll be really, I tell my students that the only thing we know right now is that the bacteria are breaking all the rules we thought existed. So it'll be a really interesting next couple of decades for food safety.
Thank you so much.
Elizabeth and Newbold and Andrea Etter, thank you both very much for your talks today. Appreciate it very much. Our next Cells to Society seminar, I believe, is March 9th, and it's from 11 a.m. to 12, and it's going to focus on microbiology and molecular genetics with Matthew Wargo as one of the speakers. Thanks for joining us today, Matthew. See you, everybody. Thank you.